Yeah.
Uh,
So let's start. It's my great pleasure to have my dear friend and inspiration in science, Dr. Vishva Dixit, at the closing lecture of our course. Of course, Vishva doesn't need an introduction, but his biography is so fascinating, and I would love to tell you some parts. Vishva was born in a small city in Kenya, East Africa, where his parents were both physicians. Like his parents and his sister and his brother, he went to the medical school in Nairobi, the capital city, where he faced the beginning of AIDS epidemic. So you can find much more of his biography in Google, and I recommend reading the Conversation with Vishva, published in Cell Death and Differentiation in 2019. So Vishva moved to USA to have his residency training, and he started in science at the William Fraser Lab in the Department of Pathology at Washington University. After a fruitful path, Vishva is now the current vice president of the discovery research at Genentech USA, and a member of several national and international Academy of Sciences, Medicine, and Arts and Sciences. Moreover, he is a recipient of numerous important honors and awards, such as the recent, I don't know to spelling, Vishva Medal for his contributions to unraveling the molecular aspects of disease-related cellular signaling with the aim to develop novel therapeutics. Vishva has made many prominent contributions to the biomedicine that we can find in the textbooks of biology and medicine, which include really many molecules involved in apoptosis, necroptosis, and paroptosis pathways. I can point to some such FED, caspase 3, 6, 7, 8, 9, caspase 10, HIPK3, gosdermin D, and more recently, the nidurine 1. Vishva has hugely impacted the field, not only by all of these discoveries, but also by his well-known generosity to spread mice and reagents throughout the world. So today, he is the best person to talk about the future perspectives on cell death and immune system. Thanks, Vishwa, again. And I really wish you were here today in person, but it's good to see you and your dogs there.
Thanks, Karina, for that wonderful introduction. Very warm, very welcoming. I do wish I was in Sao Paulo in person, but I'll take a rain check and we'll make it another day. But what I thought I would do today is go over very quickly the discovery of gosdermin D.
That's fine.
And
nice.
Then I will also launch into the very unexpected discovery of NINJ1 and give you some background and the thinking there. Okay, good. So I want to sort of move our attention to the year 2015, which is not that long ago, and what we knew. And that you could activate the non-canonical inflammasome or the canonical inflammasome. And you required the activation of caspase-1, or this led to the activation of caspase-1. And caspase-1 could cleave pro-IL-1 beta and pro-IL-18 to give you the mature cytokines, IL-1 beta and IL-18. I want you to remember two things, that IL-1 beta and IL-18 don't have a signal peptide. So they fall into this family of what we refer to as leaderless cytokines. So this is IL-1 alpha, IL-1 beta, IL-18, IL-33, and others. So a very important family. Now, the other point I want to emphasize is that you've heard about the non-canonical pathway and caspase 11. The non-canonical pathway talks to the canonical pathway because caspase 11 by itself is unable to proteolytically process pro-IL-1 beta and pro-IL-18. So cytokine processing is a function of caspase 1. So what we were wondering about in 2015 was an age-old problem in cell biology, which is how do cytokines that don't have a signal peptide, like IL-1 beta and IL-18, how do they get out of the cell because they can't go through the Golgi apparatus? What we did know was that both caspase 1 and caspase 11 could give you this form of necrotic death that is referred to as pyroptosis that is characterized by membrane perforations, so holes in membrane. And this allowed for the leakage of these leaderless cytokines, IL-1 beta and IL-18, into the exterior. So one mechanism, a very prominent mechanism by which leaderless cytokines like IL-1 beta and IL-18 get out of the cell is because the cells undergo this form of cell death, pyroptosis, and you get perforations in the membrane, and the cytokines are able to traverse the plasma membrane. The question was, what exactly is the nature of these perforations? So in order to address that question, we had undertaken a forward genetic screen. I won't go over the details because this is a relatively old paper, 2015, it's been published. But essentially, mice are subjected to this alkylating agent, ENU, which induces point mutations. And the dose of ENU is titrated, so each pedigree is getting about 30 to 40 mutations. And then there is these mutations are largely recessive, so there has to be a breeding protocol to get them to homozygosity. But long story short, what we do with these pedigrees is we get their bone marrow-derived macrophages, and we stimulate them with an activator of the inflammasome. In our case, we are interested in the non-canonical pathway, so we stimulate with LPS, cytosolic LPS. And we measure the secretion of IL-1 beta because that tells us if you secrete IL-1 beta, the inflammasome machinery is intact. So what we are really looking for in this mutagenesis experiment, and it's very extensive, it's up to 16,000 pedigrees now, and it's still ongoing. We are looking for the inflammasome to be broken. So it'll be broken if it doesn't make IL-1 beta in response to cytosolic LPS. So you can see here that the caspase 11 mutant, which is how we define the non-canonical pathway, doesn't make IL-1 beta in response to LPS, and therefore that's a good positive control. But you'll also see that this other mutant, ENUB11, that turned out to be Gesdermin D, does not release IL-1 beta into the conditioned media. And this was a bit of a mystery because in 2015, Gesdermin was really unheard of. We knew it was a member of a family. It had no signal sequence or transmembrane segment, and the function was unknown. So we wondered whether Gesdermin D was really required for IL-1 beta release. And so in order to do that, we knocked it out. And this is an experiment from our knockout studies, from the Gesdermin D knockout studies. And I'll just say that, remind you that pro IL-1 beta is 31 kD and mature IL-1 beta is 17 kD. So we can monitor this proteolytic event that's mediated by caspase-1 simply by doing a Western blot. Okay. Now remember, even when you have mature IL-1 beta, that doesn't have a signal peptide, but you can measure the release of IL-1 beta into the conditioned media by doing an ELISA. So now let's look at the Western blot data, and we will see that in wild-type cells, untreated, pro-IL-1 beta is the only form, this 31 kD form, that is found in the cells. When you activate the inflammasome, in this case with ATP, you now see the emergence of the 17 kD mature IL-1 beta, right? And in fact, if you do an ELISA, you can measure it in the conditioned media. So that's very clear. But look what happens when you do the exact same experiment with the Gesdermin D knockout. When you treat with ATP, you get maturation of IL-1 beta, so you get proteolytic maturation to give you the 17 kD. So that is very clear, but you don't get release. So whatever Gesdermin D was doing was that it was essential for release of IL-1 beta. Now, the other thing that we noticed was that the Gesdermin D was getting cleaved by pro-inflammatory caspases. I'll only show you the data here for caspase 11 when the inflammasome was engaged. So let's look at the Western blot again. Now we're going to Western blot for Gesdermin D. So it's there in wild type cells. It's Gesdermin D is there in caspase 11 null cells, but is absent in the two Gesdermin D null strains. So this says we are looking at the correct band. Now, this is in untreated cells. When we treat the cells with cytosolic LPS, you'll see in the wild type cells exposed to cytosolic LPS, you see a conversion of Gesdermin D. So there's less prominence of the full length band, and there's the appearance of a P30 fragment that doesn't happen when caspase 11 null macrophages are exposed to LPS. So from this, we concluded that when the inflammasome is activated, Gesdermin D is getting cleaved. And it's getting cleaved by the pro-inflammatory caspases, such as caspase 11. And in fact, we could show that using recombinant pure proteins, that if you took Gesdermin D and incubated it with caspase 11, the full-length protein, you got two major fragments, P30 and P20. And the cleavage was after an aspartic acid residue, aspartic acid at 276. So what have I told you till now? I've said that Gesdermin D is essential for the release of IL-1 beta. And it gets cleaved to give you a P30 and a P20 fragment. And the question was, is this cleavage incidental or is it essential for IL-1 beta release? And in order to address that, we made a knock-in mouse where we simply altered aspartate 276 to alanine. So now the Gesdermin that was expressed could no longer be cleaved. We isolated bone marrow-derived macrophages from wild-type cells, Gesdermin D null cells, and this cleavage dead mutant stimulated with cytosolic LPS, and wild-type cells make a lot of IL-1 beta. Gesdermin D null, the total knockout, makes hardly any, releases hardly any. But processing dead also doesn't appear to release IL-1 beta. So it seems that not only is Gesdermin D required, but the N-terminal, but cleavage to the N-terminal P30 and to the P20 fragment is also required. So for IL-1 beta to be released, Gesdermin D has to be cleaved. And so it dawned on us that maybe one of these fragments, P30 or P20, is the fragment responsible for making holes in membranes, for perforating the membrane, to allow IL-1 beta to be released. So we did a simple experiment. We just overexpressed full-length Gasdermin D, the P30 or the P20 fragment, and we looked at the uptake of a nuclear dye. And you can see that P30 disrupted the plasma membrane, so that you now saw very effective uptake of a nuclear dye. And in fact, when you overexpress P30, you see a great degree of cytotoxicity. So Gasdermin D is required and cleavage is required, and of the cleaved fragments, it's P30, not P20, that appears to disrupt the membrane. Now, we wondered at this time what was the mechanism of membrane disruption. And it appears that P30 oligomerizes to give you a pore. We showed this by using purified liposomes, incubating the liposomes with full-length Gasdermin D and caspase 11 so that we would get generation of the P30 fragment, and subjecting the liposomes to negative stain electron microscopy. And you can see what was very evident, and in fact very rapid, was the emergence of these pores-like structures. And we calculated this to be 24 P30s coming together to give you a pore of about 15 nanometers internal diameter. And we were very anxious to look at this at an atomic scale, but we were scooped by a number of groups on that. And but I think the structural work that has been done, probably the best by Hao Wu, is consistent with our model that comes from the biochemistry and the negative stain electron microscopy. And at the atomic level, it appears that the full-length Gasdermin D occurs in an auto-inhibited form. And when you cleave between the P30 and P20, there is a large conformational change in this N-terminal P30 fragment. There is like an unfurling, an opening up of this beta sheet in the P30 fragment that is able to insert into the membrane. And this is the structure of the Gasdermin D pore from Hao Wu's group. And the 33 P30s coming together to give you a very large pore of over about 22 nanometer pore. So I think this work solved this problem of how do leaderless cytokines come out of the cells. And it essentially said that there is a molecular machinery that leads to the generation of the N-terminus of Gasdermin D that forms a pore in membranes that allows for IL-1 beta and IL-18 and others to come out. So this then is a sort of a summary of the work in cartoon fashion, that the pore that you get from Gasdermin D is large enough that about 20 nanometers or so, that the 4.5 nanometer IL-1 beta can actually physically cross that barrier. And of course, over a certain threshold, if you have enough Gasdermin D pore formation, you collapse the electrochemical gradient and you have membrane lysis. And we measure membrane lysis by looking at the release of large molecular weight molecules, intracellular molecules such as LDH. Now, this was published by ourselves and by Feng Xiao back-to-back in 2015. And I think the work generated sort of a cottage industry. There's been a lot of interest in Gasdermin since then, and barely a week goes by before there's a publication implicating it or a family member in some process of inflammation. But I wanted to bring your attention to this recent paper from a group at Harvard, nothing to do with me, but they found strong evidence that bacteria have Gasdermins. And that, in fact, the Gasdermin gene is on an operon that also has a caspase-like protease. and that on phage infection, the operon is activated, the caspase-like protease cleaves bacterial Gesdermin D, and the N-terminal fragment of bacterial Gesdermin, I should say, the N-terminal fragment of bacterial Gesdermin makes a pore, much like the P30 pore in the bacterial membrane, killing the bacteria and depriving the phage of a replicative niche. So this is quite fascinating because, as you know, when you think of bacterial innate immunity, you think of CRISPR, you think of restriction enzymes, they're very powerful weapons against phage, but they're not conserved in you and I. So we don't have CRISPR or restriction enzymes. But here's a mechanism, the Gesdermin mechanism, that is conserved in bacteria and us. So it's quite remarkable that once evolution finds a mechanism that works, it does sort of copy and paste. Okay, so there's a lot of interest in this pathway because potentially it could be of great therapeutic benefit because, you know, through this Gesdermin pore are going to come out a lot of highly pro-inflammatory substances, not only the leaderless cytokines that I mentioned, but also pro-inflammatory substances like HMGB, ATP, etc. So if one could develop an inhibitor to Gesdermin D, then in one fell swoop, you would inhibit this sort of cytokine storm that occurs in many cases of severe inflammation. So now I want to sort of turn a page and tell you about the discovery of NINJ1. And this is really a surprising discovery. I call it a bonkers discovery, like it was so unexpected. We are still grasping, still grappling with what it means. But I'll share with you the data here. So, you know, we do this forward genetic screen with these mice, and now we have thousands, as I said, 16,000 strains. And we stimulate with LPS because we're interested in the non-canonical pathway. And we measure IL-1 beta. And by and large, the vast majority of the pedigrees are going to make, almost all of them are going to make very large amounts of IL-1 beta because we are giving a considerable stimulus with the cytosolic LPS. And because of that, you know, you have pore formation, you collapse the electrochemical gradient, you have osmotic forces at play, and the cell lysis. And we measure cell lysis by measuring LDH release. And every time we see IL-1 beta, we see LDH release. So it's just a sanity check. It's just a double check for us. Then one day they came to me and they said, look, we have a mouse strain NINJ1 that is releasing IL-1 beta, but is not releasing LDH. I'm like, that doesn't make sense to me because if it's releasing IL-1 beta, that means it's making Gesdermin D pores. If it's making Gesdermin D pores, that means at the dose of LPS we are using, the cell is dying. So I said, is the cell dying? They said, yeah, you know, whatever we do, MTT, etc., it's dead, but it's not releasing LDH. I said, well, hold on a minute. Because LDH is how we measure cell lysis in the clinical lab, in the hospital. It's how we measure cell lysis in the experimental labs. In the research labs, you can buy a kit for LDH release. And now you're telling me that you have cells that have died, but are not releasing LDH. I said, that's not possible. Now, one thing I should say is that IL-1 beta is small and LDH is large. But still, I said, I really, honestly, respectfully don't believe the data. let's make a knockout. Let's not go with this mutagenized mouse. Let's make a knockout and make sure that this crazy data idea is for real. So we made two knockout strains that are shown here in red, and the wild type are in open bars. And what we're going to do is we're going to stimulate first with LPS and measure IL-1 beta. And you will see both the wild type mice and the two NINJ1 knockouts release IL-1 beta. So that means that the Gesdermin D4 must be fine. So we were, in fact, just to confirm that, we have an antibody that only recognizes this N-terminal P30 fragment, the pore-forming fragment. So we could see after LPS treatment, it was there in the wild-type macrophages, and it was there in the NINJ1 knockouts. So NINJ1 knockouts made the Gesdermin D4 just fine, and it released IL-1 beta. But now let's look at LDH release. Wild-type macrophages that released IL-1 beta released LDH, but NINJ1 knockouts released IL-1 beta but did not release LDH. And I was just like, wow, this is really crazy. So I'm like, what is NINJ1? I never heard of this. So again, just like Gesdermin D in 2015, it was a very unexpected discovery. It's a protein of a small protein, 152 amino acids, but it's a membrane protein. It's in the plasma membrane, and it has an unusual architecture. N-terminus out, C-terminus out, and two transmembrane domains. But also in the exterior is an amphipathic helix. Okay, so you have external amphipathic helix, first transmembrane, second transmembrane, C-terminus. So it's an integral plasma membrane protein. But it's also conserved, highly conserved in evolution. In fact, Drosophila, the fly, has three ninjas, ninj A, B, and C. And it's widely expressed. So it's not some unusual cell type. It's highly expressed in myeloid cells, but it's expressed in other cells, epithelial cells included. So I should let us actually look at these cells, see what they look like. So now we are going to take bone marrow-derived macrophages and wild type. We're going to treat with nigericin. And this is your classical death that you see, necrotic death. The cells are going to first balloon up, and then they're going to collapse. Okay, now let's just see that movie. So the cells are ballooning up, and then they collapse, right? So it's like you take a balloon, prick it with a needle, and it just collapses onto itself. And this is the wild type cells, again, treated with nigericin at lower magnification, so we can see a larger field of cells. And you'll see they all expand. And then they pop and they shrivel, where they become smaller. Right? Now let's do this experiment with the NINJ1 knockouts. Okay, they expand, they expand, they expand. They expand. You can see these expanded cells, this huge, massive bubbles, but they don't pop. They don't lyse. This is overnight. So it's not like they'll lyse an hour later. No, this is overnight. And these balloon cells, the NINJ1 knockouts, are dead. I won't show you the data, but this is published in our 2021 paper. So like, wow, this is crazy. And you could say, well, hold on, maybe it is something to do with you're just measuring LDH, something crazy about LDH. But if you look at this, an unbiased analysis where we look at the LPS-induced secretome, all the proteins that are released, we see that there's a very large number of proteins whose release is compromised, is reduced in the absence of NINJ1. Okay? But here's the other important fact, is that you could say, well, this is unusual, but it has to do with Gesdermindy. But what we realized is that all roads to death, other than physical, other than freeze-thaw or microwaving cells, other than physical, all other roads to death lead to rupture that is mediated by NINJ1, lysis that's mediated by NINJ1. It's a powerful statement, but let us look at the data. So here we are measuring LDH release. Open bars are wild-type macrophages. Red bars are NINJ1 knockout macrophages. In the absence of any treatment, you've got about 18% basal release of LDH. If you freeze-thaw the cells, then of course there's no difference between NINJ1 knockouts and wild-type. Nigericin, I just showed you, there's a dependence on NINJ1. Streptolysin or Listeria other pore-forming toxins. To get lysis, you also need NINJ1. But look at this. You're killing cells with cisplatin, a chemotherapy. And that is also dependent. The lysis is also dependent on NINJ1. So all roads to biological death that are where one sees cell lysis, that is mediated by NINJ1. Now, but this is a very surprising finding, because when you were in high school, you were taught by a biology teacher that when cells die, then osmotic forces are responsible for their ballooning and bursting, right? So when cells die, they balloon and then they burst. That's what you were taught in high school. That's a basic concept of biology, right? But what I'm saying is that your high school teacher, junior high school teacher, was wrong. That in fact, this step, cell death and cell lysis, can be decoupled. And that cell lysis and the release of large molecular weight entities is actually an active process that is mediated by NINJ1, genetically mediated. So this is very surprising. In your career, you often get to challenge work that is in medical textbooks or immunology textbooks, but you don't often get to challenge your high school biology teacher, right? So this is very, very unusual. So now we have this very unusual molecule, NINJ1. So again, if you go from the N terminus to the C terminus, it has this extracellular amphipathic helix, transmembrane domain, and second transmembrane domain. If we look at conservation in this conserve score, lower the score, higher the conservation, you'll see that this amphipathic helix is highly conserved from fly to man. And if you do an alanine scanning, so we mutate 5 amino acids at a time, we walk down the molecule altering 5 amino acids to alanine at a time. And then we read out their ability to kill. So wild type cells that are overexpressed are going to kill the cell. This is just 293 T cells. But you'll see that as you walk down the N terminus, it is quite tolerant of mutations. But as soon as you mess with the amphipathic helix, you disable the ability of NINJ1 mediated lysis. So the amphipathic helix is very important. So one of the questions was, what happens when we look at endogenous NINJ1? And we do this, again, in bone marrow-derived macrophages, treat with nigericin. It's a time course, 0, 1 hour, 2 hour. This is a NINJ1 knockout, so make sure we are looking at the right bands. And you will see that in the absence of nigericin, so these are in healthy cells, NINJ1 is mostly monomeric, slightly oligomeric, mostly monomeric. But within an hour in this blue native page gel, you see evidence of polymerization. You're getting very acute polymerization. And you can, in fact, see this by immunofluorescence in the untreated cells. NINJ1 on the plasma membrane is more diffuse. In nigericin-treated cells, it's more focal, punctate, as if it's coming together. And it's, of course, absent in the knockouts. We are looking at the right stuff. So we're wondering what allows NINJ1 to go from here to here. So when cells are dying, you go from monomeric form to polymeric form. How does that happen? So we really looked for signals that maybe NINJ1 is getting cleaved, maybe it's undergoing phosphorylation, maybe it's undergoing ubiquitination. But no, and none of that turned out to be the case. So what we think, and I'll get back to this at the very end, is that NINJ1 sits in the plasma membrane in healthy cells, and it's monomeric. When cells start dying, no matter what the reason they die, they're going to lose energy, they're going to lose ATP. And remember that to maintain plasma membrane composition, asymmetry, tension, you need to expand energy. So in a dying cell, the health of the plasma membrane is altered. We think the NINJ1 senses this alteration, and this alteration triggers its oligomerization. And I'll come to this concept later. But it doesn't appear to be a post-translational modification. So now you have to ask yourself the question, well, why would evolution conserve such a mechanism? I mean, it almost seems like it's trying to make a dead cell deader, right? I mean, the cell is dead. Why does it care if it lyses rapidly or slowly? Why would it care? I mean, one possibility is that we have cells are dying within our cells all the time. And but it's an immunologically silent death because those dead cells are engulfed by neighboring cells or by the reticuloendothelial system, something we refer to as apherocytosis. But if you exceed that ability to clear cells, then the cells will lyse. So like apoptotic cells will undergo secondary necrosis. They will lyse, and they're going to release mediators of inflammation, which will alert or activate the innate immune system, saying there's something wrong here. We have exceeded the capacity of clearance. Cells are dying. It could be due to an infection, for example, an acute infection. And the released contents of the cells, being pro-inflammatory, being filled with the damps, serve a useful function because they attract the innate immune system. But there's also, if you agree with that, there's also a therapeutic consequence, which is that if you were able to clamp the dead cells so that they didn't lyse, then in situations of sterile inflammation, this may be helpful. So, sterile inflammation is, of course, where a pathogen is not involved, but there you don't want excess inflammation. And if you could clamp dead cells from lysing, you would give the neighboring cells more time to engulf them. Okay. So you would say, well, this hypothesis makes a couple of predictions that if you lack NINJ1, then you are more susceptible to infections. And that's sort of, these data, though published, are still preliminary. We see that with Citrobacter rodentium. If we challenge wild-type mice, they do fine. But NINJ1 knockouts, they do much more poorly. Work from Igor Brodsky's lab shows the same data with Yersinia pseudotuberculosis. So it's consistent with the notion that if you don't have cell lysis during an infection, you don't mount an optimal immune response. And I'll be the first one to say that we haven't studied this in any further detail. When I say optimal immune response, we haven't measured cytokines, we haven't measured pathogen load. So there's a lot to be done there. But what about models of sterile inflammation? And in order to address that question, we not only wanted to use our knockout animals, but we were all still attracted by this idea of generating a potential therapeutic. So we wanted an antibody that would inhibit the function of NINJ1. And this just got published this week, so I won't belabor you on the details, except we immunized NINJ1 knockout mice with extracellular vesicles that highly expressed NINJ1. So this was a very good immunization strategy to get a very broad spectrum of antibodies. And these antibodies were screened for their ability to block membrane rupture. And you can see here, we were very fortunate. We got one antibody, D1, that was almost as good as the knockout in inhibiting membrane lysis. Okay. And this antibody maps to the very C terminus. Okay. So this antibody very potently inhibits cell lysis. If we just look at LDH release, the black bars here are the NINJ1 knockout. The blue bars are cells treated with the antibodies. And on the x-axis is the insult. In this case, it's cytosolic LPS. But you can see that the suppression of death is comparable between the knockout and the antibody treated. Right? So this antibody really does inhibit the function of NINJ1, the lysis function. And in fact, when cells are exposed to this antibody, dying cells, you still get these balloon-like structures that are retained, just like in the knockout, NINJ1 knockout. So it's really blocking NINJ1 function. And we think it blocks NINJ1 function by blocking this polymerization. So if we overexpress NINJ1, purify it, and subject it to negative stain electron microscopy, we see these very filamentous structures, sort of beads on a string. And in some cases, they form complete circles. If this was a pore, this would be like 10 times the size of the Gesdermendy pore, maybe almost 100 times, actually. Very, very large potential pores. But if we do this in the presence of the D1 antibody, then there's no evidence of filament formation. Another question was, what is the best sterile injury model to look for a possible therapeutic benefit? And in this, we were guided by some GWAS data from the UK Biobank that showed that there are polymorphisms in NINJ1. And these polymorphisms are associated with very low levels of liver enzymes, so AST, ALT, and LDH. And of course, as you know, liver enzymes is how we measure liver disease. And for this reason, it may not have been the best reason, but buttressed by this knowledge, we decided to look at liver injury models. So the first one we looked at was very severe apoptosis model, TNF and D-galactosamine. This spills over into secondary necrosis, so you get cell lysis. The first experiment we did was with the knockout, the genetic model. And if we just look at the wild type hepatocytes, this is a section through a liver, and staining for NINJ1, you will see that NINJ1 is actually quite prominent on the plasma membrane. And this is an inducible knockout. The details don't matter, but in the knockout strain, the staining for NINJ1 is gone. So we've got a good system, a genetic system to test. And now if we look at markers of liver injury, let's just look at LDH. When you treat them with TNF and DGAL, the enzymes LDH, ALT, AST all go through the roof. But if the mice are null for NINJ1, then you see there is a substantial inhibition of release of these markers of liver injury. So this is with the genetic model. What happens when we do this with the antibody? And here we pre-treat with the antibody and then give TNF and DGAL. And you will see that the antibody-treated cells are shown in blue, same experiment. And you will see that there is a great degree of suppression of liver enzymes. And I would say, in my mind's eye, even more impressively, you see that circulating markers of inflammation like serum IL-18 or HMGB1 are suppressed. So it seems that this antibody is suppressing liver damage and generalized emergence of markers of inflammation. It's acting as an anti-inflammatory. Now we can do this same sort of study with other liver injury models. This is a Conné model. It's really CTL killing. You'll see in the genetic model, these are the dark black circles, there's considerable protection. In the antibody treatment, there's also considerable protection. We, working with Ben Steinberg and Jan Mossman at University of Toronto, we did yet another liver injury model. This time is ischemia reperfusion injury. This is with antibody treatment. And again, we see attenuation in markers of liver damage and attenuation in markers of inflammation like neutrophil recruitment. So I think this raises the possibility that NINJ1 may be a druggable target. That, as I said, that cell death is normally immunologically silent because we have this very powerful buffer of phagocytosis, which takes up dead cells before they lyse and before they release the DAPs. They're taken up. But if you have excess of cell death, then you have this secondary necrosis. You have the release of intracellular DAMPs. This propagates inflammation. But this antibody suggests that if you can inhibit, the cells are still going to die. That's not coming back. But if you can inhibit the lysis, then you give a longer opportunity for engulfment by phagocytes and neighboring cells, and that may be the mechanism, anti-inflammatory mechanism. So this was a very surprising finding, right? And whenever you make surprising findings, there's a lot of disbelief, which is fine. There should be. There's a lot of healthy skepticism. So we published the first paper in 2021, and we really wanted to do the atomic structure of NINJ1. But it was very difficult to convince people to do it. But fortunately for us, a group in Switzerland has done the structure of NINJ1. Again, it's published this week in Nature. It's both our paper and the Swiss paper or advanced online publications. But their data, I'm happy to say, really at an atomic level shows the NINJ1 pole. And so it's very satisfying to see that. But I think neither their paper nor our work to date answers a centrally important question, which is what I alluded to earlier, which is what is the mechanism for activation of NINJ1? How does it go from that monomer to a polymer? I raised the possibility that maybe NINJ1 is monitoring membrane health, tension, composition, partner. We really don't know. And that is something we are very interested in working out. So let me thank the people who actually did the work. This is a long-standing collaboration with Nobuhiko Kayagaki and his team, Irma Stowe, Bettina Lee, and Ofer Kornfeld. The antibody was made by Ryan Kiley in our antibody generation group. Animal models were run by Wayne Lee, outstanding animal modeler, and an equally outstanding pathologist, Josh Webster, who helped with these studies. But really the forward genetics approach is a very large undertaking that's undertaking with the Australian Phenomics facility that's based in Canberra, large group of people that is led by Ed Bertram and Chris Kudna. So with that, I'll stop and be happy to take questions. Okay,
oh my gosh.
Wow.
Tony, my gosh. Always finished your talks without words and so anxious. I don't know, it's a mixture of feelings. Oh my gosh, so excited. Of course, I'm looking forward to have this D1 clone to test in our infectious models here. And I open for questions because I have some of them, but we wait for the students first.
I have multiple questions as well.
So wait. Abolade, you can start.
Okay.
Guys, you are so lucky to have this kind of lectures this week, especially this one.
Yes. To me, I would say that lucky is an understatement. We should look for more words to, qualify this. I'm so happy to, to be here. So I have just one question. I don't know if. So my question is, you majorly spoke about the interaction or the interaction of the caspase 11 to pro higher 1 beta, and also it's in relation to gasdemin or gasdemin. So what about caspase 1?
Yes, so caspase 1 will cleave gasdermin D. So caspase 1, 4, and 5 in man. And remember, 4 and 5 is represented by 11 in the mouse. So it's a bit confusing, but caspase 1 and 11 in the mouse, they will also cleave gasdermin D. But neither caspase 4, 5, or 11 will cleave IL-1 beta or IL-18. So for IL-1 beta, IL-18, you have to activate caspase 1.
Okay. Thank you.
Oh.
Thank you, Abolade. Lucas?
Hello, can you hear me?
Yep, loud and clear.
Okay, sorry if there's some noise in the background. I'm in laboratory, so. In one of your when you were showing a immunochemistry, immunofluorescence for Ning, Ning1, and I was a bit, I was, I got a bit confused because I understood that Ning1 is expressed in the cell membrane.
Yeah.
But I also saw in the Western blot result that there is a change of size to this protein.
Yes.
And I don't know if that may be like, if it's related to, no, it might be a really stupid question actually, but
No.
I don't know if there is a translocation from the cytoplasm to the membrane, to the cell membrane, or if it's related to the difference you saw in the immunofluorescence results when you showed with that in one of the images it was sort of like small dots, and then you had condensed dots, and I don't know if maybe it is related to the same thing you saw on the blot results.
Yeah, no, that's a very good question. So remember the Western blot, we ran a native page gel. So there you're going to see proteins as they migrate by size. So it's not a denaturing system, it's a non-denaturing system. So non-covalently associated proteins will run at a higher molecular weight, right? Or will run slower. So it's a native gel. And cells are migrating there as a function of their size. Okay. With regards to whether Ning1 is a plasma membrane protein exclusively, our staining shows some intracellular background. And so we've sort of tried to pursue that biochemically with cell fractionation. And I would say the vast majority of Ning1 is probably in the plasma membrane or is in the plasma membrane, but there's probably some in the intracellular membranes, Golgi. They could just represent the fraction that is being transported to the plasma membrane. But now Ning1 happens to be part of a family. And there are members of Ning1 like TM160, which is also like an orphan protein that is there on the inner mitochondrial membrane. So I think as this field will expand, people will probably start finding, just like Ning1 is on the plasma membrane, maybe Ning1-like proteins that are on other membranes, because maybe there is a need for all membranous structures to resculpt themselves. There may be what I think of as the formation of apertures or remodeling. And we've tried to look at TMEM160, but we haven't been able to find a function for it as yet. But I suspect that the field, somebody will. But I suspect this family has this sort of function in other organelle systems.
Oh, I get it. If I may, just the last one. If I'm not wrong, you need cell rupture as a signaling for the cell that's not dying through apoptosis to get to be engulfed.
Yeah.
So if there is no cell rupture and it's not dying through apoptosis, what signal will that be then? Because the cell can't be there. Like, it's not like you're trying to undie the cell. You're just trying to avoid more damage. No, if you mean like in terms of trying to treat the cell, trying to inhibit Ning1. If you're trying to do that, and the cell is not now getting cell rupture, how then in in the organism, is the cells going to be found and eaten by microphages, for example? I was really confused by that because it's not apoptosis. So
right, but I think it'll still express phosphatidylserine on the membrane, so it'll still express eat me signals. So the dying cell. So
yeah, that was my question.
So that's and I think we have to be very careful that as researchers, we, for the sake of simplicity, we catalog pathways and we start thinking of them as unique, separate entities. But in reality, as a pathologist, when a pathologist sees cell death after injury, let's say ischemia reperfusion injury, which is sort of a classic sterile injury you would see after stroke or myocardial infarct, there is a mixture of cell deaths happening. There's certainly an apoptosis. There's certainly necrosis. So, and necrosis has its various forms. So I think that you don't have pathological injury in the real world that gives you a singular form of cell death. But remember, when you talk of apoptosis, you're talking about a kilogram of cells disappearing from your body every day, right? Now that is, of course, a very efficient physiological death, not pathological death, physiological death, where the reticuloendothelial system is primed to take up apoptotic cells. But when you have injury, whether that is injury by viruses or bacteria, or injury, sterile injury, ischemia reperfusion, you really have a mixture of deaths, right? We would like to believe, we would like to say, I've developed an apoptosis inhibitor, and it's going to work in ischemia or stroke or heart attack. And in reality, those have all been mixed results because the pathology is always mixed in that injury is always a mix of pathologies in terms of cell death.
Oh, okay. Well, thank you very much for your answer and for your time. And it was a really great presentation. I've said it before, but it's a new world for me. I've just recently gone dive deep in this cell death world, and it's been amazing. All the presentations, but yours is one that was really, really, really good.
Oh, thank you.
Thank you very much.
Thanks, Lucas. Ricardo?
Well, 1,000 questions, but just
No, Ricardo,
to,
one,
so,
Gustavo, one, and me, one.
I know,
and,
Karine. I'm not going to monopolize. I'm just buying two questions then. It makes just one and I'll be quiet. So what happens to the cell if you leave it there? Will eventually collapse?
Eventually,
and what happens to the mice? Are they just fine?
Yeah, so two good questions. So eventually the cells will burst. I mean, in our hands, probably after 24 hours, they will eventually lyse. There is a paper that says 48 hours. So I think the good news is the field is picking up on this and is repeating the study. So it's between 24 hours and 48 hours, which in the is a lifetime for a cell, you know. So, but remember, in the mouse now, physiologically, you don't see really anything, because if cells are dying, they're taken up. It's not like in culture, you're not giving them a chance to undergo secondary necrosis, right? And so now the Ninja knockout have the severe problem. It is variably penetrant, but severe hydrocephalus. And we don't understand that. That's why we've gone to an inducible knockout system now. So, because it just breeding became a problem because we were just losing so many mice to hydrocephalus. Yeah, but I know nothing else about that.
I
I'll
will skip.
wait.
Well, I skip you because my question was exactly related to the central nervous system. You told us that ninjurin 1 is expressed in many cells, including epithelial cells, but I don't remember to hear to see any paper about the ninjurin 1 expression in central nervous system cells.
Yes, so it's
And
been reported, ninjurin was first reported as a gene that came up on nerve injury.
nerve injury.
Nerve
Yes,
injury.
yeah,
It's also been
yes.
reported as a p53 inducible gene.
Yes, you're right. I remember now. Yes.
Yeah, I think it is regulated. We ourselves have not really studied the regulation. We haven't repeated the nerve injury studies, for example, but there is a considerable literature of nerve injury, and that's how it got its name.
And because I was thinking about the relation between the role of paroptosis, because now there are several papers demonstrating the role of paroptosis in the central nervous system, not only neurodegenerative disease that we know, but also during the neurodevelopment and of course, now related to depression and anxious and other neuropsychiatric disorders. So I wonder if they revisit these papers of Gazermin D knockout mice with the ninjurin 1 knockout.
What we are doing is that really for CNS studies, the rat is a much better model, especially if you want to do stroke, ischemia, reperfusion, which is probably what we want to do first. So we've just created a NINJ1 knockout rat. And so we are going to do ischemia, reperfusion. So both heart, both myocardial infarct and stroke in that model. I mean, that's the sort of easier, lower hanging fruit. I mean, the neurodegenerative diseases, that's a complex area, as you know.
Yes.
If there's promise there, then I think it would allow people would be more interested in exploring it further. But I think that's where we're going right now.
Thanks. Gu?
Hey, Vishwa. Your work has been really inspiration and challenge at the same time all through your career. Let me just ask a quick question first, because I think I missed, and then I'll ask a more general question. So is NINJ1 needed for necroptosis and for the CTL killing?
Very good question. So for necroptosis, the requirement for NINJ1 is less. Still required, but it's less. And I think that's because MLKL can play a compensatory role, but it is still required. We've done those. I think what is surprising is that nobody has shown formally that MLKL makes a pole, right? So that's eluded, but there's no structure of MLKL pole.
Right.
So, and then CTL killing, we just haven't looked.
You haven't done?
No. There's so many questions like, NINJ1 is very highly expressed on neutrophils. So people say, well, what about neutrophils, netosis or neutrophils? Yes. We haven't looked. There's only so much one can do. So that's why we're giving the mice away, everything like usual. So people should look. It's a lot of unanswered questions.
But we have these mice. They're very good. We have these mice that you brought, these mice, but it is so hard to cross. To breed, yes, to breed
so
them.
You should ask for the inducible knockout. Maybe that's the
one
knockout.
Well, then we're gonna do that,
yes.
Because Vishwa, I haven't been able to fill out the MTA as well. We are trying to get to append to our previous one. And I have to work out that. But if you think the inducible would be easier, then I think then it would be best. And Karina and I will get
Yeah.
that.
It'll be much easier. And, when you append the MTA, you can copy me. So,
Yeah.
Okay.
We'll do that. Okay, so now the real thing that it's really puzzling, and you mentioned why the NINJ1 is so important and it's evolutionary conserved. Why the need of popping up a cell, even a cell that has pores, and releasing the pro-inflammatory and is being dead. So it puzzles me a little bit. And
it's very puzzling. And my thought is, so, Drosophila have 3 ninjas, A, B, and C. We've knocked out, we as with a collaborator, we don't work on flies, but they've knocked out 2 of them just in the intestinal epithelial cells of the fly. Okay, so you can do that. And then those flies are very susceptible to intracellular pathogens. So I think that cell lysis, very rapid cell lysis, is important for defense against intracellular pathogens, because then you very quickly throw the pathogens out into the extracellular space that is filled with complement and neutrophils and other. The last thing you want is for the pathogen to be retained. That I have to say is speculation.
But that would make sense if the pathogens could still, grow in the corpse,
yes.
The cell corpse. Okay. And so do you know if any pathogens can still grow or be protected in a way in the cell corpse?
Absolutely no evidence. It's a very good question. I wish but I think it raises the question that you are asking. You're saying that if this is a possibility, then show me in cells like the NINJ1 knockouts that the intracellular pathogen lives longer.
Right.
But I think those studies now can be done, possibly in the fly system, which is, a purer system. But it's an open question, because I am still very surprised at the conservation and evolution of this process.
Another possibility is I don't know, but maybe some dumps are so important to induce this kind of inflammation, because it's hard to believe, because others such as IL-1 beta and IL-18 and other.
But I think in support of your idea, Karina, is that, when you get pancreatitis, which can be fatal, the damp that's being thrown out by the cells is trypsin. That's what's going to kill you, right?
Yeah.
So I think as immunologists, and I had this conversation with Seamus, because I said, large molecular weight damps, and he said, oh, you can't say that. And I think we get very fixated with terminology, because if you've got pancreatitis, you forget about IL-1 beta, IL-1 alpha, IL-18. It's what's killing you. Protease, the trypsin that's being released, right?
And even HMGB1, I don't remember well, but I think the knockout mice, the HMGB1 knockout mice is so susceptible to many of
it could be HMGB1
infection, and also resistant to sterile inflammation and pathologies and so on.
Yeah.
I don't remember well, but I think
so. So I think, I showed you that proteomics data. There are a lot of proteins whose release is compromised.
Yes.
So,
Yes.
and so
So,
I think formally what you raise is a possibility, because people say, well, you can't call them dams. I okay, call them mediators of inflammation, right?
yes.
Just like I said, trypsin is a mediator of inflammation and
Inflammation.
pancreatitis. In an
Mhm.
immunology textbook, you're not going to see trypsin as a damp, right? They'll say, oh, it's not a cytokine. But in the real world,
But it is NINJ1 dependent,
yes,
maybe.
yes,
Yes.
it
Well,
is.
Yes.
but then this is not evolutionary force. This is a pathology that now you can use, your knowledge to counteract.
Right. So, Gustavo, very important point, which is that all of these systems develop in evolution because of the host pathogen interactions and race. So, I think Ninja as you know, evolution only cares about you till you reproduce, and after that, you're useless, right?
Right.
So, yeah, and so I think these mechanisms that have been defined, like Gesdermindy or Ninjuan or whatever, I think they're the core of why they exist is in the host pathogen race. So that's why we are entertaining this possibility of intracellular pathogens.
No, that thing I like more.
Mhm.
The dams, it's a bit weird to me because I think in the paroptosis, the gasdermine pore is enough to alarm the system.
But
So I don't think that extra dumps would interfere too much. And then when you have necroptosis, you said that NINJ1 is less important because eventually the other pore. So I like more the idea of, get rid of any possibility of pathogens to grow or to hide in a cell corpse, because the immune system will not be, that I like more.
I think it's it may have developed that way, and it may have a consequence in
But I have another question more related to your teacher in the school teacher, okay, which is more like maybe physical. So normal membrane bilayer would not rupture by the force of osmotic force. This is this like a physical thing? And then, now nature develops something that would help because this bilayer would not, not be so sensitive. Why I'm asking that? Actually, what I'm asking is I'm trying to think of this other way around. I'm thinking that, let's say that the nature develops something else to hold a membrane, because we need this membrane to be intact. Okay, that's very important for the unicellular and the multicellular organisms. And then it might be important for this membrane to rupture, so you create a second layer of regulation. So my question is that, have you ever looked at binding partners of NINJ1 in the membrane? So is NINJ1 actually there to prevent, to help? But if you don't activate NINJ1, so this other thing will hold the membrane until there's no energy left. And then that's where you see in 24 or 48 hours.
That's exactly what we are thinking. And so we were able to, fortunately, do a fairly clean proteomics experiment. We have a few candidates that came down quite stoichiometrically. And so we are looking at them right now. So, yeah, I mean,
All right.
I'm
But
quite,
there will be a second part of the story.
yeah, but that's exactly the approach we are using. So,
Okay. So it's not that, because if the membrane was easy to burst, then as a physical, then you don't need anything. It would just burst by osmotic thing.
right.
And Ninja One is something that facilitates, which
Absolutely. Accelerates. Yeah.
accelerates. So that means that something else must
Yeah.
be in place as well.
Yeah.
All right. I like that.
Yeah.
Okay. I'm done with questions for a moment.
You got my annotations and you read for Dixit, because we think the same stuff. But let me just complement one question that Gustavo did in terms of kinetics.
Yeah.
So when you quantitate IO1 and IO18, it's quite the same level of release. But what about the kinetics? Are you trying to read in early points to see if in early points there is the difference?
So it's a good point. We haven't looked at kinetics. There may well be an influence on kinetics. And I think the other question that we haven't entertained in our liver injury model, we treat with the D1 antibody and we see an anti-inflammatory effect. As I just said, there are cells like neutrophils that express NINJ1 highly. What if we were inhibiting neutrophil function in those models? So I think there's a lot of work to be done mechanistically because there is a big distance between the cell culture experiments and the animal experiments. And even though we like to relate them in our model, I would say that we don't have all the data to make, I think these are all possibilities. I don't think we have a firm model for the anti-inflammatory function of this NINJ1 antibody. It could well be a participant inhibiting lysis. It could be inhibiting neutrophil function. It could be all of those that lead to this benefit that we see. But I think that needs to be dissected out.
Yeah, because from the concept that I have, and maybe it's outdated, is that you have the Gazarmin D pore and IO1 and IO18 will go through it. But then you have the rupture of the membrane, and then you have a much bigger release of this cytokines. So when you don't have NINJ1, then you release a little bit. And with time, you accumulate.
Of course, yes.
Kinetics is different.
Kinetics,
maybe different.
Well, it's a good point, Ricardo.
And also the production, right? Because if the cells are still alive, I don't know if the name is, but if the protein synthesis is still going on, maybe later on you'll have even bigger.
I think kinetics is a good point. Yeah.
Cool.
All right.
Thanks.
I think abology has a
abology.
Yes.
Abology, you're from Nigeria?
Yes.
Yes.
Do you know any Portuguese?
I'm learning. I'm learning Portuguese.
You're learning Portuguese?
Yes.
Okay.
He's in my lab, Vishva, doing PhD.
Okay.
Yes.
You're teaching him Portuguese. Excellent.
Yes. Thank you.
He understands quite a bit now. It's been a year.
Yeah.
Okay.
So I just have one quick basic question, because about NINJ1, you said they mostly reside in the plasma membrane, and they also contribute to plasma membrane rupture, right? So that means they are inflammatory. So my question is there any interplay between NINJ1 and TRIF or the toll-like receptors in general?
is there any interplay between toll-like receptors and NINJ1?
Yes.
Yeah. Well, certain toll-like receptors under certain conditions will lead to cell death, and if you engage them, and in those roles, NINJ1 will have a role. But we have not looked whether NINJ1 is sort of inducible by TLR engagement, for example. So we have not looked at that, at the regulation of NINJ1 by toll-like receptors.
Okay. Thank you.
So let's finish and let Vishva have his lunch, because now, yes, it's lunch time there. Vishva, thanks a lot. Of course, we keep in touch, and we will arrange our next.
Thank you so much.
Yeah, absolutely. And we hope to see you in Sao Paulo soon. South Africa first.
Well, we will meet first in
South Africa,
but then in São Paulo. Definitely leave some weeks in June free, and I'll get back to you as soon as possible. Right?
All right, guys.
Okay, super thanks.
Super
Send our love
thanks.
to Manju,
Vishva.
the whole family.
Yes.
Bye.
Bye-bye.
Bye.
Thank you. Ok, guys.
Ô,
So
Gustavo,
now-
só uma coisa. Tem mecanismo sim o tempo inteiro de reparo de membrana. Tem até um paper do grupo do Doug que mostra lá que esse reparo mantém a
o
expressão
LAP
de...
lá.
Não LAP. LAP é internalização, né? É o
Não,
outro
é o SCORT.
mecanismo.
Não, SCORT.
SCORT 2. Que
É.
você tem o tempo inteiro essa membrana sendo reparada por esses pequenos...
Sim, mas não quando tem choque osmótico, é muito estranho, porque, entendeu? A
Não,
célula
choque
aí
osmótico vai romper. Como ele mostrou lá que também...
não vai romper. Ele
Não,
freezer
choque osmótico
é
não,
outra
é o único
coisa.
que não precisa. No choque
Entendeu? Choque
osmótico, a NINJ1 explode igual.
Ali é
Choque
freeze
osmótico.
and
No
thaw.
freeze and thaw.
Não é choque
É
osmótico.
verdade.
Mas ele não falou que
Não.
quando você
Mas
tem
você
célula
tem um
nocaute,
choque. Você tem um
você
choque
tem.
osmótico, Ricardo, quando a célula faz os furinhos, entendeu? Então a coisa toda que eu acho que tá aí dizendo esse segundo capítulo aí é que a NINJ1 não está sozinha. A NINJ1 está lá porque tem outra coisa que a gente não sabe, que está segurando a célula para não ficar o tempo todo estourando. Fez um furinho, fez uma coisa, ela
Mas é
explode.
isso. O SCORT 2 é um desses mecanismos de quando você faz um
Esse é
furinho
o
lá e
reparo.
consertar.
Então, esse é o reparo, mas deve ter uma coisa física, porque essa coisa da NINJ é física, a princípio, né? Anyways, mas vai ser muito interessante, pessoal. E é isso. Espero que vocês tenham aproveitado.
Acho que o mais importante, assim, gente, é que eu gostaria mesmo que tivesse tocado em vocês, né? Depois de tanto tempo, tantas descobertas tão impressionantes e tão importantes, né? Em todas essas vias de morte celular, ele continua tendo o brilho nos olhos, ele continua falando, oh my gosh, olha esse resultado aqui, meu, que inesperado, eu quero entender isso, sabe? Então, assim, eu de verdade gostaria muito de olhar nos olhos de vocês e que vocês, especialmente nessa semana, porque a gente passa, né? São altos e baixos, né? Mas que essa semana tenha sido assim,